I am starting a series on cancer. I am giving my introduction. I am professor in Department of Chemistry, MMH College. I have done my PhD in chemistry and have learned biotechnology in the Indian Institute of Science. After that, I have worked in National Institute of Immunology on drug delivery system. Title of my talk is Cancer Not to be Feared, and understanding the cancer through history, causes and development. First and foremost thing about cancer is not to be feared. Half of the problem of the cancer or the disease gets doubled moment we start worrying too much instead of focusing on treatment. So before going into detail, let's first know the history and evidences available about it in the literature. From the dawn of history, evidences and literature is available about cancer. Oldest evidence about tumor cells like growth in bones is available from Egyptian mummies, though different word was used at that time, not cancer. An Egyptian medical textbook called the Edwin Smith Papyrus on trauma surgery describes disease not treatable. The Edwin Smith Papyrus is an ancient Egyptian medical text named after Edwin Smith, who bought it in 1862. Papyrus is a writing material used in Egypt in ancient times, and since paper used to write these materials were made from plant Cyperus papyrus, family Cyperaceae, hence the written material was also called papyrus. This is a slide which showing the leaflet of that particular book. Word cancer originates from scientist Greek physician Hippocrates' work. He noticed tumor cells looking like crab and named them carcinoma. It is a Greek word for crab. Later on, Latin word cancer meaning crab like zodiac sign was used for cancer cells. He noticed that there is a central body to a tumor and the tumor extensions appeared as the legs of the crab. The veins stretched on all sides as the animal crab has its feet, from where it derives its name. Hippocrates named the disease cancer when he dissected a woman's breast tumor and saw tendrils of it spread over the breast tissues in a way that reminded him of the legs of a crab. Later on, scientists found different type of cancer, and not necessarily all type of cancer cells looks like a crab. It's important to note that all type of cancer cells, they doesn't look like crab. This is a picture which shows the similarity of the cancer cells with a crab. I mean, here you can see the central body and the veins coming out of it as a crab has a legs. This is name of scientist tells us, a Roman physician. He translated the Greek term carcinoma used by Hippocrates into Latin word cancer meaning crab. Cancer comes from the Greek word carcinos or crab. It refers to the claw-like extensions into neighboring tissues. This is the page of the book written by Celsus. This is another book which was written by Galen, a scientist. He was Greek physician. He used the word onkos, Greek word for swelling, to describe the tumors. Now the question is how to define the cancer. What is cancer? Simplest definition of cancer was given by the American Cancer Society. Cancer is a group of diseases characterized by uncontrolled growth and spread of abnormal cells. If the spread is not controlled, it can result in death. So far, whatever we know about the cancer, evidences available. So here is the modern knowledge about cancer on the basis of knowledge we gained so far. Common term which are being used for cancer: cancer, neoplasia, tumor, neoplasm, all these words are commonly used for cancer. The word cancer comes from the Latin, originally Greek, derived term crab, because the way a cancer adheres to any part that it seizes upon in an obstinate manner like the crab. Hippocrates first described cancer as having a central body with a tendency to reach out and spread like the arms of a crab. Besides the popular generic term cancer used by the most people, there is another more technical term, neoplasia. Neoplasia means, neo means new, plasma means tissues, or neoplasm literally means new tissues in Greek. This indicates that cancer are actually new growth of cells in the body. That is very important point. These are the new growth of cells in the body. Terms such as mass and lump, they are also used to describe any overgrowth of tissues. However, These terms may not necessarily mean that such growth contain cancer cells. It is also a very another important point. Another term for cancer is malignant tumor. Tumor literally means swelling or mass. In this case, it refers to a mass of non-structured new cells which have no known purpose in the physiological function of the body. Type of cancerous growth. There are two general types of tumors: benign, which are non-cancerous tumors, and malignant cancerous tumors. A benign tumor is composed of cells that will not invade other unrelated tissues or organs of the body, although it may continue to grow in size abnormally. Now, types of cancerous growth. A malignant tumor is composed of cells that invade the basement membrane and invade or spread to other parts of the body. This occurs either by the direct extension to neighboring organs and or tissues, or by metastaticizing to distant sites by means of the vascular system, the blood stream, the lymphatic stream, or by seeding or implantation of cancer cells in the body activities. Now, types of abnormal cell growths. In addition to neoplasia, there are several other terms referring to abnormal cell growth. These include the following: hyperplasia, which refers to abnormal increase in the number of cells which are in a normal component of that tissue and are arranged in a normal fashion with subsequent enlargement of the affected part. One example is the thyroid hyperplasia, an enlargement of the thyroid gland caused by abnormal rapid growth of the epithelial cells lining the follicles. Another example is guitar stringing leads to hyperplasia of the cells on the thumb. A callus is formed. The callus on the thumb is a hyperplastic growth. Hypertrophy refers to the abnormal increase in the size of each cell. For example, the increase in cell size of cardiac muscles. Metaplasia refers to the replacement of one mature cell type with another mature cell type. For example, squamous metaplasia of the respiratory column epithelium, as evidenced by the metaplastic cough of a smoker. Dysplasia refers to the replacement of one mature cell type with a less mature cell type. For example, dysplasia of the cervix epithelium. Hyperplasia, metaplasia and dysplasia are reversible because they are result of a stimulus. Neoplasia is irreversible because it is autonomous. So this is a very important difference. These other mentioned hyperplasia, metaplasia and dysplasia, they are reversible because they are results of a stimulus. But neoplasia, which is for the cancer, is irreversible because it is autonomous. This is a picture of a cancer cell and a normal cells. Normal cells, one can see, generally have a smooth boundary, and here cancer cells have a rough boundary. Here you have a clump of cells and the nucleus in the broken, it looks they are broken. Similarly, this is another picture of normal cells and the cancer cells. Here the cancer cells looks very rough boundaries, you have extension like amoeba, but the normal cells are smooth. This slide is showing in situ cancers and invasive cancers. Right side it is an invasive cancer, and where you can see the cells is spreading to the other part of the body and spreading to other cells. Shape of normal cells versus the cancer cells. Cancer cells are always like a clump. The right picture which you can see, and in the left picture you can see where the cancer cells are like a mass. Other than in the blue we are showing the normal cells. Normal cells in all the pictures have a normal boundary and a smooth boundary. This is a tumor terminology generalization. What happens that for tumor there are certain patterns in the scientific word to define it, how to name it. So names of benign tumors usually end with the oma, regardless of their cell type. For example, a benign glandular tumor which is of epithelium tissue is called adenoma, and a benign bone tumor is called osteoma, while a malignant glandular tumor is called adenocarcinoma and a malignant bone tumor is called osteosarcoma. The difference is here in the second part we are referring to the type of cells also. In addition to benign tumors, there are in situ tumors and invasive tumors. In situ tumors do not invade the basement membrane, whereas invasive tumors do invade the basement membrane. What are the characteristics of cancer cells? First thing, they are the abnormal cells. Cells are the structural units of all living things. A human body has trillion of cells. All body functions are performed by healthy cells, just like doing all household things. If a person is not well, he or she cannot do various jobs in the house. Similarly, unhealthy cells cannot do various functions. Cells makes it possible for us to carry out all kinds of functions of life, e.g. breathing, respiration, digestion, sleeping and walking, etc. However, some cells, they stop functioning or behaving abnormally and cannot serve any useful purpose in the body at all, are known as cancerous cells. Another important characteristic of cancer cells are invasion. Normal cells respond to signal from the other cells which tell them that they have reached a boundary. Cancer cells do not respond to these signals and they extend into nearby tissues, often with finger-like projections. Another important characteristic of cancer cells is uncontrolled division of cells. The most fundamental characteristic of cancer cells is their ability to reproduce themselves. They do this simply by dividing. One cell becomes 2, the 2 becomes 4 and so on. The division of normal and healthy cells occurs in a regulated and systematic fashion. This is a big major difference between a normal cell and a cancer cells. Cancer cells keep dividing and there is no control in the body over that. So in same thing in uncontrolled division of cells, in most part of the body the cells continually divide and forms new cells to supply the material for the growth or to replace worn out of or injured cells. For example, when you cut your finger, certain cells divide rapidly until the tissue is healed and the skin is repaired. They will then go back to their normal rate of division. But cancer cells, they keep dividing. There is no check in their division. In contrast, cancer cells divide in a haphazard manner and the result is that they typically pile up into a non-structured mass or tumor. That's why you always detect the cancer by seeing a mass or lump or a tumor in the body. What are the techniques which have been used for the development of cancer? Actually, in the scientific world, every scientific branch in the world gets the boost, when the technology keeps developing, they keep adding to the particular field. So similarly, the cancer and its development in the field was also helped by many techniques which kept coming from time to time. First report is of famous Scottish surgeon John Hunter. He suggested that some cancers might be cured by surgery and described how the surgeon might decide which cancers to operate upon. If the tumor had not invaded nearby tissues and was movable, he said there is no impropriety in removing it. Another technique which was later developed was anesthesia. Anesthesia allowed surgery to flourish and classic cancer operations such as the radical mastectomy were developed. Similarly, in the 19th century, they saw the birth of scientific oncology with the use of the modern microscope in studying the diseased tissues. Rudolf Virchow, often called the founder of cellular pathology, provided the scientific basis for the modern pathologic study of cancer. Now you have some other techniques also, like the development in field of pathology. Moragani had linked autopsy finding scenes with the unaided eye with the clinical course of illness. Virchow correlated microscopic pathology to illness. So these people, they developed the technique, and with the help of these techniques we got the more knowledge about the cancer. Similarly, other techniques are also there. This method not only allowed a better understanding of the damage cancer had done, but also aided the development of cancer surgery. Body tissues removed by the surgeon could not be examined and a precise diagnosis could be made. This pathologist could also tell the surgeon whether the operation had completely removed the cancer. Field of pathology has done the significant contribution in the development and the knowledge about the cancer. What are the main causes of the cancer? This is very important question. There are several theories given from time to time about how the body gets the cancer. Humoral theory, lymph theory, blastema theory. Similarly, there are so many other theories, but we should find scientific reasons behind it, and that's why scientists continued working on finding scientific reasons. For example, what are the genetic and environmental causes, or the lifestyle, or any other risk factor causing cancer? First scientific reason is viral and chemical carcinogens. In 1950, Kotsushiburu Yamagiwa and Koichi Ishikawa at Tokyo University, they induced cancer in the lab animals for the first time by applying coal tar to rabbit skin. John Hill of London recognized tobacco as a carcinogen, a substance known or believed to cause cancer in humans. Today, we recognize and avoid many specific substances that cause cancer, like coal tars and their derivatives like benzene, some hydrocarbons like aniline, a substance used to make dyes, asbestos, and many others, ionizing radiations from a variety of sources, including the sun. It is also known to cause cancer. To ensure the public safety, the government has set safety standards for many substances, including benzene. These are the safety standards, and government has decided which chemicals can be used in any process or which chemical should not be used, which can cause cancer. Another scientific reason which strongly help you to find out the cancer causes are the genes and the genetic causes. Scientists already knew that cancer could be caused by chemicals, radiations, and viruses, and that sometimes cancer seems to run in the families. But as the understanding of DNA and genes increased, they learned that it was the damage to DNA by chemicals and radiation, or the introduction of new DNA sequences by viruses that often led to the development of cancer. So there are two main type of genes which they have been studied well, and they are known to cause cancer. They are called oncogenes and tumor suppressor genes. During the 1970s, scientists discovered two particularly important family of genes related to cancer: oncogenes and tumor suppressor genes. Normal genes slow down the cell division, repair DNA errors, and tell cells when to die and how to die, a process which is known as apoptosis or programmed cell death. When tumor suppressor genes don't work properly, cells can grow out of control, leading to cancer. Functioning of both the genes can be compared with the accelerator and brake system of a motor vehicle. Oncogenes becomes active and they behave like an accelerator of a vehicle. Too much acceleration causes vehicle to go out of control. For it to work properly, there need to be ways to control how fast it goes. A proto-oncogene normally functions in a way that it similar to an accelerator, which helps the car speed to be regulated and pick up speed. Malfunctioning of oncogenes causes the cells to divide out of control. A tumor suppressor gene is like the brake pedal on a car. It normally keeps the cell from dividing in too quickly, just as a brake keeps a car from going too fast. When something goes wrong with the gene, for example, if a mutation causes it to stop working, cell division can get out of control. Slowly, medical scientists are identifying the oncogenes and tumor suppressor genes that are damaged by the chemicals or radiation, and those that, when inherited, can lead to the cancer. For example, 1990s discovery of two genes that cause some breast cancers, BRCA1 and BRCA2, is a step forward because these genes can be used to identify people who have a higher risk of developing cancer. If these genes are present, BRCA1 and BRCA2, there are chances that a person will definitely get the cancer at some stage of his or her lifetime. These genes cause cells to grow out of control and become cancer cells. They are formed by changes or mutations of certain normal genes of the cells called proto-oncogenes. Proto-oncogenes are the genes that normally control how often a cell divides and the degree to which it differentiates or specializes in a specific function in the body. Now there are some common questions which are related to cancer, which the people usually ask, and because there is a lot of confusion about the cancer in the society. First, because people immediately, if you hear the name that somebody is suffering from cancer, you are scared. So you need not to be scared. It seems everything causes cancer. This is a very common question, but the answer is no. Not only those things which damage our DNA and cells multiply uncontrollably only cause cancer. Similarly, there is another common question related to cancer, because at present we know there are several types of vaccines. Especially in India, we have the BCG vaccine from 1950, and after that we have a smallpox, we have the polio vaccine, and now recently during the pandemic we got the corona vaccine. Is there any vaccine has been developed for the cancer? Yes, HPV vaccine. It protects against the human papilloma virus, HPV. If this virus stays in the body for a long time, it can cause some type of cancer. The FDA, US Food and Drug Administration, has approved HPV vaccines to prevent these type of cancers: cervical cancer, vaginal cancer, vulval cancers, and anal cancers. So these are the references which one can go through if they want to know the details, and from where we have taken lot of data and lot of pictures. Thank you.